Materials and method. My experimental species is Heteronistas fossilis. These fish are collected from
Let's
the.
keep this type.
These are the chemicals, which is fipronil, and the IUPAC name is plus minus 5 amino 1,2,3 dichloro alpha 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 trichloro pitolil 4 trichloro methyl sulfonate pyrazole 3 carbonitrile. There is no available literature regarding to fipronil, so we should determine the lethal concentration of LC50. First, we take 1 mg per liter concentration, then 0.1 mg per liter concentration, then no one fish died, then 0.2 mg per liter concentration. 2 fish were died, and the mortality percentage is 33.33%. When 0.3 mg per liter concentration were put in, then 4 fishes were died, and the mortality rate is 66.66%. And after 0.4 mg per liter concentration, there 5 fishes were died, and the mortality rate is 83.33%. These are
NC50.
NC50. These are the graph from which we calculate the LC50 of fipronil. The LC50 is 0.257 mg per liter, in which half of the fishes were died. So 3 fishes were died from 6 out of the 6 fishes. These are the experimental method. First, we collect the animal, the Heteronistas fossilis. Then we narcotize this animal by using chloroform. Then tissues were collected, and brain and kidney, and those tissues are kept in the wound fluid for fixing. After 24 hours, these tissues are was paraffin.
Okay. We'll skip this slide because we know it. Everyone know it mostly. I think this is normal histological procedure. If to skip this slide, come to your result.
This is the result, and this is the slide of control phase. There is optic septum, which is present in the brain of Heteronistas fossilis, and neuroglia, which is the special cell which protect the neurons and epithelium cells.
This is a 40x.
40x. These are the slides of after treated 10 days and 21 days. Several changes occurs, including vaporization, necrosis, dilation of nuclei. After 21 days, there is more changes occur. More severe vaporization, severe necrosis, and pynotic nuclei also occur. And this is the histology of kidney of control fish, Heteronistas fossilis. The fishes kidney is mesonephric kidney, and is consists of nephrons, renal corpuscle, and renal tubule. Renal corpuscle is consists of glomerulus and Bowman's capsule, and the renal tubule is consists of PCT, Henley's loop, and DCT. These are the DCT, glomerulus, and Bowman's capsule. These are the slides of third treated. There also shows many changes, including vacuolization, degeneration of renal tubule after 10 days, and after 21 days, there is also changes. Many changes occur, including necrosis of interstitial cell, dilation and shrinkage of Bowman's capsule, formation of vacuole, hypertrophy of distal convoluted tubule. These are the discussion in the brain and kidney after the treated by the sublethal concentration of fipronil. Many changes occurs, including vacuolization, vacuole formation occur, necrosis, very many cells damaged, pinoptic nuclei, nuclei are become shrinkage, and several changes also occur in the kidney of the treated fishes. including dilation of renal tubule, hypertrophy of distal convoluted tubule, and fecolation also occurred. Necrosis, very many cells were damaged. Acknowledgement. I special thanks and gratitude to my supervisor, Dr. Kuranjit Das sir, and my classmate Abhijit Das. Thank you.
Okay, thank you. You asked question. Open for discussion. Is there any question? Well, who did it? That. Okay, thank you. Thank you very much. Next participant is Queen Soikia, Mongoldoy College, Mongoldoy. Oh. Yes. The next participant is Queen Soikia from Mongoldoy College. The topic is targeting of NOGCGMP, CGMP pathway, in silico investigation of isopyridine in the management of erectile dysfunction.
Next speaker.
Queen Soikia.
That's it.
So please come, Odhitsmita Sharma, Bhattacharya University, Bajani. A comparative analysis of oyster mushroom cultivation and yield on various substrates.
One below, sir. Good afternoon, everyone. Respected teachers, all the guests present over here, and my dear juniors. Myself, Madhusmita Sharma. I have completed my bachelor's in botany from Tihu College and now pursuing master's from Bhattacharya University. So first of all, I would like to thank Tihu College Science Forum for giving me this opportunity to present my dissertation topic from final semester of bachelor degree, that is a comparative analysis of oyster mushroom cultivation and yield on various substrates. So oyster mushroom, scientifically known as Pleurotus ostreatus, is a group of edible mushroom and are known for their rapid growth, nutritional value, and adaptability. Talking about the benefits of consuming oyster mushroom, it has nutritional potentiality such as it contains 85 to 95% of moisture, 35 to 70% of carbohydrates, 15 to 34.7% of protein, 10% of fat, 6 to 10% of mineral. Then it also contains antimicrobial properties. It contains ethanolic properties, and then it also contains anti-tumor properties. Contain beta glucan, show a competitive regulation in colon cancer cell line. Then, talking about the antioxidant properties, it also contains phenolic flavonoids, glycoside polysaccharide, which act as antioxidant. Moreover, it also contains antiviral properties, it contains beta glucan, polysaccharide, phenolic compounds, and enzyme inhibitors. Then economical values of oyster mushrooms is such wide that according to database market research in global oyster mushroom, global oyster mushroom market was valued approximately 60 billion US dollar in 2024 and is expected to reach approximately 110 billion by 2031 with CAGR of 7.8%. Then, talking about the environmental values of oyster mushroom cultivation, the whole process of cultivation of oyster mushroom was we use substrate nothing but the agro wastage, which also denotes the waste recycling. Then low carbon footprint, it's connected with the soil health improvement and reduced use of agrochemicals. Then, talking about materials and method, the first step was collection of substrate. We collected the substrate from the local area of Tihu. Then we cut it into small pieces and then boil it. This was the first stage of sterilization and also to smoothen the structure of substrate. Then we sun dried it to remove the extra moisture and then we used 70% ethanol for the final sterilization process. And, the next process was preparation of PP bags. We make the PP bags in a cylindrical form so that mycelia can grow in all the possible directions. And we layered it with the substrate and the sponge in alternative manner with 3 to 4 layers. Then coming to the placing of bags, we place it in a room of our botany department where there is minimal to no sunlight, which is suitable for mushroom growth. And we waited for 3 to 4 days, I guess, for the mycelia growth so that we can water it. Coming to the result varies with the different substrate. As we know, all the different substrate provide a different kind of nutrition to the growth and development of mushroom fruiting bodies. Then, as you can see in the slide, the rice straw provide the highest yield, followed by mustard straw and then wood flesh. And talking about the lowest yield, water hyacinth, rice has dry leaves and banana leaves almost showed zero yield.
So you create second different substance.
Yes. Then talking about biological efficiency, rice straw showed the highest one, followed by mustard straw. And once again, water hyacinth, rice has dry leaves and banana leaves showed the lowest one. Then identifying a suitable substrate for mushroom production is an important task for improving food security. Days for mycelia invasion, days for pinhead formation, and the fruiting bodies formation. Overall yield, Pleurotus ostratus mushroom was significantly affected by the substrate tested. As we can see, thus the rice straw substrate gave the highest yield, followed by the mustard straw and substrate respectively. Therefore, this substrate could be used as potential mushroom production.
For mushroom production in the study area.
The references,
These are the references. You can check it out.
So last but not the least, I would like to
I
thank
would like
each
to
and
thank
every
each and every
faculty member of Department of Department of Botany, Tihu College for giving
College for
us
giving
this
us
opportunity
this
and
opportunity
motivation
and
throughout the
motivation
journey.
throughout the journey.
Joy to Tihu Mahavidyalaya. Thank you very
Tihu
much.
Mahavidyalaya. Thank you very much.
Thank you very much. The topic is open for discussion. Is there any question? Otherwise, your teacher will ask a question. Will ask you.
So do you have any new ideas apart from this that should continue this work? It came any question to you? Well, we already know that the rice flow will be yielding the heights. And
Sir,
prove it.
Sir,
It is traditionally known.
Yeah.
we could mix the
we could mix the
substrates,
substrates like
70% of rice straw and
70% of
then
rice straw
30% of
and then
other
30% of
substrates.
other
We
substrate.
could test
We
it
could
out, but
tested it out, but
due to lack
due
of
to
time.
lack of time, we couldn't
Okay. But
raise anything.
did it give some idea, new idea? You thought like you could have mixed the
About
substrate
substrate,
which it came first and second one. Something like that. Suppose the
sir,
second one, what substrate it was? First is rice flow.
The second one, mustard
second
straw.
one,
Mustard straw. What if like you could have combined this rice straw and mustard straw together?
That's what I was
That's
saying.
what I was
Sir,
saying.
we
Sir,
couldn't do
we
it
couldn't do
because
it
of lack of
because
time,
of lack of time, and
it was risky.
it was
So
risky.
we,
Okay, you
So
continue next. Okay.
thank you very much.
thank you very much.
Okay, thank you. Next participant, I once again announce the name Queen Soitia of Mongol College. I think she's not here. Next one is Gitanjoti Kodita. And his topic is small indigenous species of hill of Himalayan region in Bakka district, Assam, and its conservation status. Ishan Jyoti Kodita. Ishan Jyoti Kodita. The next participant is Krishnangi Goswami, Bhattadev University, Bajali, and her topic is anti-asthmatic ethno-medicinal plants used by the Bodo tribe of Pakska district of Assam.
কোনোবাই ভাত নোখোৱাকৈ আছে নেকি? ভাতটো খাই আহিলে ভাল হ'ব, অলপ দেৰিকৈও লাগে। সেইফালে বন্ধ হৈ যাব খোৱা তাৰমানে। ভাতটো খাই লওক কেইদিনত। এইটো আৰম্ভ হোৱা নাই যদি অলপ ব্ৰেক কৰি দিওঁ, ভাতটো খাই লওক 10 মিনিটত। তুমি খালা ভাত খাইছিলি? খাই লওক, ভাতটো খাই লওক। হ'ব আমি এইখিনি চাই আছো। খাই লওক, সিফালে বন্ধ কৰিলে ভাত খাব নাপালে আমাৰ বেয়া লাগিব নহয়। 10 মিনিট হে, 10 মিনিট পিছত খাই পেলাই ডাইৰেক্ট আহি যাব। একো নাই, এইফালে ৰেডি কৰি দিব ছাৰে। ভাত খাইছা তোমালোকে? ইয়াতে খাইছে। গেছখিনি আছে। সেইফালে ভাতটো বন্ধ কৰি দিব। ইমান দূৰ আহি ভাতটো নাখালে বেয়া লাগিব। থাকি যাব ইয়াত থাকি যাব। একদম মাটি পাৰ আছে ইয়াত সবাই কৰা। So আমি এতিয়া ইনক্লুড করতে
উত্তৰলৈকে আহিব নাই?
হ্যাঁ
তাতে আহা। অনলাইন এইটো অনলাইন হ'ব নেকি?
Hello. No. গরম মানে বাইরে চেয়ারপারসন হবে কিন্তু আর বুঝতে পারবো কথা বলছে নন ইনভেসিভ
Tihu বুলি আছে নহয়? হেড কোড আছে নহয়? এইটো হেৰি অলপ হেড কৰি থ'ব পাৰি।
এতিয়া ৰিমুভ কৰি দি দিব স্যার Good afternoon, everyone. I apologize for breaking up the session, and that session will be continued after another session that is session 14. Now we are going to start our session 14. For this session, I request our Bulbuli Kanikar madam to share the session, and we have two invited speeches. First speech is going to delivered by Puranjit Das, Assistant Professor of Botanical University. He will talk on the topic use of non-invasive sample in wildlife genetics. And before going to present his presentation, I would like to thank him and going to first of all, we are going to felicitate Mr. Gamosa. I request Megha Chatri to felicitate. A memento and a sapling. Thank you, sir. Now I hand over the schedule to our chairperson, Imani.
So, good afternoon. First of all, respectfully, I want to say that the chairperson, Dr. Bulbuli Kanikar, my faculty members of this college, and their participants. Today I am going to deliver a lecture on use of non-invasive samples in wildlife genetic studies through microsatellite marker. So, in wildlife genetics, non-invasive samples are used basically because we can't use invasive samples because it will definitely harm animal. And in wildlife particularly, we cannot reach the animal to take samples invasively like blood samples or any other samples. So non-invasive samples is preferred in this case. So what is non-invasive samples? The definition is that non-invasive samples are collected without capturing or harming the animal. Without harming the animal, the samples are collected. So what are the samples? Examples. So here, feces, feather, urine, saliva, sweat, skin, excess. Excess মানে যেটো excess আমাৰ পাওঁ আমি calcium to সবাটো নহয়. Excess, that is the residual embryonic tissues are there. এটা কি এটা excel ভিতৰত সেইখিনি আমাক লাগে. Environmental DNA মানে DNA যিখিনি আমি those DNA which are found in environment, in soil, in water, these are called environmental DNA, and these are non-invasive samples, and these samples are collected during the So these non-invasive samples are collected, and importance of these non-invasive samplings are it minimizes stress on wildlife. There is no effect on wildlife while collecting the non-invasive samples. So second one is it reduces risk of injury or mortality. As the samples are collected from the environment, there is no risk of mortality or injuring the animal. The third one is it's suitable for rare, elusive, or endangered species. In some times, only few individuals of a species are there in wildlife. So we can't take this sample from such type of individuals or species. So in those cases, we must take the samples from non-invasive samples. So this is called reduces suitable for rare, elusive, and endangered species. So next one is it enables long-term monitoring. So wild animals are moving from one place to other, so it is suitable for that also, and it supports ethical research without injury, without harming the animal. This is called ethical research. So what is wildlife genetics actually? Genetics, we know genetics, all of us know genetics, but what is wildlife genetics? Wildlife genetics is the study of genetic variation within and between wild animals population, using DNA analysis to understand biodiversity, track movements, manage conservation efforts. So what is that? That wild animals, we can study wild animals interspecific as well as interspecific. That is differentiation between the species, differentiation within the species. Then next, it is important for assessing genetic health of populations, which affects their resilience to threats. Resilience to threats means threats to become future threats like climate change, and help inform strategies for species identification, disease monitoring, and combating wildlife crimes. Next one is key applications include creating genetic reference database, monitoring population health, developing genetic-based solutions for captive breeding, and habitat restoration and future conservation strategies. We can achieve these goals by applying these genetic principles in wildlife animals. So next one is techniques used in wildlife genetics. So several techniques used in wildlife genetics. First one is data and DNA analysis. So studying an animal's DNA to understand its genetic makeup. Then non-invasive sampling, collecting samples like hair, feces, and others. And then bioinformatics. These are the steps. Bioinformatics, after nucleotides, we know the nucleotide sequence of the DNA. Then we have to analyze it, and this analysis is done by using several bioinformatics tools. Next is the DNA barcoding, common DNA analysis method. What are the methods? So DNA barcoding is one of the methods that where mitochondrial DNA mainly sequenced. And then second one is short tandem repeats analysis. Mitochondrial DNA can be analyzed from the samples. Then short tandem repeats can be analyzed for samples like non-invasive samples, and single nucleotide polymorphism (SNP) analysis is done for nuclear DNA from the collected samples which are degraded one. So in the current communication we have made that microsatellites, about what I want to say about microsatellites. What are those? The microsatellites are repetitive sequence of DNA consist of short tandem repeats of 1 to 6 base pairs. And the number of repeats at a specific locus can vary greatly between individuals and between populations also. So it is polymorphic. It can be effectively used for intraspecific variation. So they are highly variable among individuals. This inherent variability makes them exceptionally powerful genetic markers for a wide array of ecological and conservation applications, particularly when dealing with the limited and often degraded DNA from non-invasive samples. We obtain DNA from non-invasive samples, and those DNAs are from, for example, from feces or from hairs, are degraded DNA. We generally got degraded DNA. That is, DNA is not exactly found as in the case of normal DNA, that is the living animals, DNA collected from living animals. So what are those microsatellites? The microsatellites are called codominant markers. So microsatellites are codominant markers. Why they are called codominant marker? We know that there is a condition of homozygous and heterozygous, and microsatellites can be analyzed through homozygous or heterozygous condition. So microsatellites, this allows researchers to distinguish between homozygous and heterozygous individuals. Highly polymorphic it is. Another characteristics of microsatellite, a high degree of polymorphism means that many different alleles exist within a population. And third one is versatility in application. Microsatellites are ideal for individual genotyping and identification, kinship analysis. Individual genotyping, we know it, individual kinship analysis to find out their nearest one, their friends, their family, friends, etc. Family basically. Estimating population size, assessing genetic diversity and bottlenecks, and studying population structure, gene flow, and dispersal pattern and hybridization events. All these can be examined, all this can study by using microsatellite marker. So microsatellite markers are dominant and codominant markers. For dominant markers, particularly RAPD, ALP used, and for codominant markers, microsatellites and RFLP markers are used. Then why microsatellites for non-invasive samples? Why microsatellites used for non-invasive samples? These are the causes. Sensitivity for low quality DNA. So DNA we obtain from fecal DNA, suppose it is low quality DNA. So we can't sequence larger portion of DNA, that is nuclear sequence. So in case of microsatellites, we can sequence it because these are small fragments of DNA. You can see here in the below portion that fragments are there, CTTT, 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 like this repetitions. In the first part, 10 repeats are there, and then in the second part, 18 repeats are there. They are of same species, but there are different repetitions of these microsatellites. And these repetitions, by examining the repetitions, number of repetitions, we can differentiate an animal. Animal means individual. These are the two individuals of from same species, but this allele is repeated 10 times, and whereas second allele one is repeated 18 times. In this way, we can find out the variation among the individuals of a particular species. So I have an experimental evidence regarding the microsatellite markers. In the studies, I have used Rhinoceros unicornis as my experimental animal, and I used to find out, take samples from Kaziranga National Park, Rajiv Gandhi Orang National Park, and Pavitra wildlife samples, and we basically used dung samples. Samples from fecal samples from herbivores are called dung samples, and it is difficult to obtain DNA from dung samples because dung samples contain sometimes DNA of fragments of DNA. We can sequence DNA from 10 parts also. So it is difficult to obtain particular DNA of that particular animal. So in the first method, how dung samples were collected? In the first method, dung samples were collected by directly monitoring the animals. And in the second part, in the second way, fresh dung samples were also collected from the field. So all these dung samples were preserved in 95% alcohol, and then these dung samples were used to take extract DNA from these dung samples, and isolated DNA was amplified using 6 microsatellite loci, RH1, RH3, RH5, RH7, RH9, and IR10. All these are microsatellite loci. So these are the primer sequence for these microsatellite loci, and third one is annealing temperature. Then the standardization of primer. I used 10 ml microliter reaction for primer standardization for PCR reactions, and PCR standardization was made through gradient PCRs, and we used negative control reactions for dung samples. That is DNA obtained for dung samples. So all these, after collecting data were set in the software microsecker, and some bioinformatics tools are also used. These are Excel microsatellite toolkit. Various data format were prepared by using convert program, then GenPop, Harlequin, structure, genetics, Genelix, structure harvester, bottleneck, etc. All these are bioinformatics tools. These are the tools I used in the experiment. So allele frequency, generally what is, it is only for students also only. What is allele frequency, you know it, Hardy-Weinberg law, p square plus 2q plus q square. How allele frequency is calculated, I will not discuss it here. So example of allele frequency, this will also not discuss. Then come to the result section, where microsatellite alleles and their frequencies in RNA, rhinoceros unicornis from 3 populations were found. Then this is the chart. I will not describe all these things. Then this is allelic richness. Allelic richness is a measure of genetic diversity that quantifies the number of different alleles. That is gene variance at a given genetic locus within a population, and this allelic richness is presented here for 6 different locus. Then population pairwise FST, it was calculated by using Kimura 2 parameter from the 3 rhino population, and the estimated population, the estimation is done by using some software, and comparison is made like this. Then Rhino Kaziranga 0. It will definitely 0. Rhino Kaziranga, Rhino Povitora, it will 0.54. Rhino Kaziranga, Rhino Orang, no differences. Rhino Povitora, Rhino Kaziranga, differentiation is found in minors. Right, this is 0, and Rhino Povitora and Rhino Orang, there is a differentiation, little differentiation. So like this, we FST calculated, and it is presented in the table. So the range 0 to 0.0 to 0.05 to consider as little genetic differentiation, and 0.05 to 0.15 indicates moderate genetic differentiation, and 0.15 to 0.25 indicates great genetic differentiation. So genetic diversity per locus, this is my findings in the 6 different loci from 3 different wildlife areas. These are the mean number, that is genetic diversity per locus. I found it. Number 1 is 0.69, second one is 0.66, and third one is 0.64. And genetic gene diversity refers to the variety of alleles present at a particular locus or across multiple loci within a population. So come to the next. This is the graphs of allele frequencies. The larger one means they have more frequency, and smaller, shorter one is less frequency, having less frequency in the microsatellite loci. Microsatellite loci are given in the like this one: RH1, RH3, RH5, RH7, RH9, IR10. These are microsatellite loci, and these are the frequency. This is highest frequency is about 7.7. It's exactly 6.9. So findings on rhino populations. Non-invasive sampling using microsatellite marker is a powerful and ethical approach in wildlife genetics. It supports conservation by providing critical data without disturbing animals. So I have already discussed it. The microsatellite data using analysis in 3 rhino populations show probably of gene flow among the population. It is the findings that there is a flow of genes. Flow of genes means flow of animals from one habitat to other. Then the third one is the FST. FST people show the differentiation between the rhino population of Kaziranga and orang population. So this is one of the most important findings, that rhino population of Kaziranga and rhino population of orang, there is a differentiation, genetic differentiation between the two populations. So application of genetics in wildlife management. This is general topic. This is not my one. General topic, general discussion that genetics plays a pivotal role in effective wildlife conservation and management. A genetic approach ensures scientifically sound strategies for preserving species and ecosystem. These are the points: identifying distinct populations, managing genetic health, designing conservation strategies, monitoring illegal trade and poachings, and tracking gene flow between populations. Then next one is challenges and limitation of non-invasive samples. So these are the challenges and limitation. DNA degradation in non-invasive samples. Usually, while we extract DNA from non-invasive samples, the DNA samples usually degraded one. Then there is a risk of contamination. There is a risk of allelic dropout, and it requires that non-invasive samples requires optimized protocol and validation. Have to do again and again. That means challenges in conservation genetics, limited genetic data, high cost of analysis, ethical and logistic issue, and habitat fragmentation. These are the challenges in conservation genetics. Then thank you very much for your attention. So,
session is open for any questions and comments.
Sorry, I mean, again, I'm not from biology.
Okay.
What is the significant difference, I mean, for laymen between the two kinds of
significant differences.
yeah, like visible, anything visible that we can differentiate?
No, there is no visible differences. There is no visible differences. Differences in the DNA sequence only. It is useful for future of the species. Currently, but externally, more morphologically, we can't see differentiation between this.
So why is
There
this?
may be minor differences. There may be differences in hair. There may be differences in the length of hair. There may be differences in the horn. There may be differences on some other differences of these individuals, but it is very minute. We can't examine it. But genetic differentiation, if there is a differentiation, if there is not any morphologically differentiation, no morphological differentiation between the individuals, there may have some genetic differentiation because their lineage may be different. Their parents may be different.
Oh.
They may come from different lineages. So
Really? Okay. So I thought, I mean, they're the same species, just got separated.
yes,
here.
we human, we are Homo sapiens, we have differentiations. But differentiation not in gene. Differentiation basically occur in the non-genetic part, that is non-coding DNA. We have two types of DNA, coding DNA and non-coding DNA. Coding DNA is same. You have enzymes. Sometimes we have taken samples of replica samples. suppose there is a pile of dung and I have collected 2 or 3 samples from this dung. Because there is also a thought-provoking thing that rhino usually is a rhino fecal matter is usually found and they drop their piles in a particular place. So there may be so collecting samples, fecal samples from without following the animal is difficult. There may be samples from different animals, different individuals.
There is no more questions. Then thank you, sir.
Okay, thank you. Thank you.
In the big applause.
Thank you very much.
So I love you for next week. In my next piece will be delivered by Purangai Meter, Sapatulo Pinwest. Okay, yeah. So, we felicitate you for I would like to request Pratishita Das to please felicitate Dr. Puranjoy Mukund sir. Sir is from Bhattacharya University, Rojave. So, can you felicitate Sir Mr. Kambuza and a plant and a
I invited people to be.
Pointer আছে নেকি?
Pointer,
Good afternoon, everyone. Today I'm going to present a paper actually. This work is already done. It's about to get published. So before getting published, I think I will try to show a little bit how it is done. So the paper, its name is Microbial Diversity and Probiotic Potential of Fermented Beverages of Missing Tribe in Assam, integrating metagenomic profiling with ethnocultural knowledge. Although the title sounds a bit very lengthy, but it's a very simple. See, it says that metagenomics. Metagenomics means it's like the whole genome, you analyze it and you see what are the species are here. Okay, so generally we will be seeing here the fermented beverages used by the missing tribe. So we already know that the fermented foods have lots of important probiotics and all, probiotics, bacteria and all. Those are very much good for our gut health and all. So now what are those bacteria? How many types of bacteria are there in a fermented product? Or it is that only the good bacteria are there, or there are some bad bacteria like pathogens are there? And what are the metabolites they produce? There are ample number of questions like how those microbes came into that fermented product. Okay, so and in our study we have checked for even the store sample. Like if we store the fermented product for 1 year, then what could be the changes in microbial diversity? Okay, coming to introduction. As we all know that traditional fermented foods are culturally very important worldwide, as it can extend the shelf life of the product and it enhances with lots of nutritional benefit. Microbial enzyme is produced with unique flavor and having lots of health promoting compounds like vitamins, organic acid, and various probiotics compounds. So among the probiotics, we know that those are the LAB, those are the lactic acid bacteria. Those are responsible for producing all those probiotics compound and all, like lactobacillus, streptococcus. And if we go to see the diversity of ethnic food or traditional fermented food, Assam is home of various different tribes. Those ethnic tribes have their own unique way of producing their own fermented food. Okay? So each community now, based on their geographical region, based on their substrate they are using, based on their way of their production, the product of the fermented product get varies, and similarly the microbes also get varies. So it is region specific, the microbial community changes. So although our region is placed with lots of different tribes and with different numbers of fermented food, but only a few scientific studies have been done on those fermented food on very proper systematic way, like microbial diversity, probiotic level, functional metabolism, how microbial succession happened, how those microbes came to the fermented product, and what are the potential pathogens. These are not done properly. So very limited numbers of products have only been carried out such studies. So if we have that much limited study, then the problem with those food will be, it will be very much it will reduce in consistency in in taste, flavor, and how like it will be concerned for safetyness also, like at what level those product will become poisonous or it get the pathogenic bacteria will be rising. This all concern comes to the mind when you do not study very systematic. So but with the modern intervention of like metagenomics and all, it have been possible because we can now do those fermentation in a controlled environment. We can sterilize those starter, and we can practice in a hygienic way so that we can control those quality the required way. So among that, we in our study we choose the missing community. Missing community generally it practices generally these are the types of fermented food. Namsing, that is the dry fish, Ikung is the bamboo shoot, and rice beer and epop. So here generally we focus on what called fermented beverages. So we'll be focusing on apong, that is rice beer, and its starter cake, that is epop. From where we believe that from epop the microbes came to the fermented products. So this will be done by the metagenomic approach by NGS technology, which will give us all the microbial diversity and its functional potential. So our objective will be to see the microbial diversity of the fermented beverages, to check the microbial succession, how it changes the microbial dynamics. Next, to predict the metabolic function, what are the metabolites they are producing in due time. And to check the beneficiary, like what are numbers of probiotics and the numbers of harmful pathogen, how it increases and how it came. So sample collection, we collected this sample from 3 different districts, Dhemaji, Majuli and Golaghat, because those districts are prominently habited by missing tribe. And those samples were collected in triplicate order, because if we collect the rice beer, we collected from 3 different areas and we mix it together, then we send it for metagenomics. Similarly for starter cake also, we collected from 3 different districts and we mix it and we send it for metagenomics. So to understand the result, we have to follow this code. We have given the code EF1 for the fresh, that is the rice beer, which we call the has pani, or lau pani we call it. And the PA1 we coded as the store one. If we store it for 1 year, then what will happen? And EF2 will be the starter cake, that is the epop, from where it is believed that from where the microbes came to the final fermented product. Okay, just remember the code EF1, PA1 and EF2 as a starter cake. So this is the picture. You can see EF1 is the rice beer, EF2 is the starter cake, and PA1 is the 1 year old in a regular refrigerator. It's a store one, freezing. So this is the way it's prepared. the entire process, like the preparation of starter cake and the preparation of the entire rice beer. Now it is believed that before preparation of this one, they collect those medicinal herb. It says that from this medicinal herb they prepare the starter cake. From the starter cake, it's believed that the starter cake have lots of different medicinal plant, and those medicinal plant will have different endophytic fungi and endophytic bacteria. Those endophytic bacteria and endophytic fungi are responsible to enhance the probiotic level in the fermented products. Now it is in those particular plant are the bacteria or the necessary probiotic fungi are available here. So at earlier time they used to make around like 100 plus plant species and all, but now with due time, as due to various anthropogenic activities and all, plant have been lost. So only like 20 to 21 plant are regularly being used by those tribes. These are some of the common which are very much available. So the magic comes from those plants. So material methodology, I'll skip this part because we did it based on outsourcing this. But just to simplify, we collected this sample, did DNA extraction, PCR amplification, then sequencing. Then after sequencing, we did data processing. Then those data we generated based on, then we checked those taxonomic wise, and we have checked by group wise and all, and we have checked the diversity and all the functional prediction. Okay, coming to result. Now, first of all, we'll discuss only the EF1 and EF2 because those are the fresh product. EF1 is the fresh rice beer and EF2 is the fresh starter cake. These two are combined because these two are together they will produce those probiotic levels of bacteria. Now, when we sequence them, a total of 2,67,000 of sequence we got it, and we have seen that in starter cake it was dominated by lactococcus around 22%. But here, although lactococcus is also a probiotic bacteria, but the prominent, the main one, the lactobacillus was seen in a very low number. Even it didn't came in the range of 1% also. But in the final product, when you see the data, we have seen that lactobacillus was high to 65% lactobacillus. Here the magic came. So the starter cake, which was just less than 1% of lactobacillus, in the final product it comes to around 65%. And other bacteria also are there, but those are not to the probiotic level, are in the range of 1 to 3%. Okay. So this is the diagram, pie diagram. Here we can see EF1, you can see lactobacillus, it is 65%, lactobacillus is 21%, and in case of EF2, it's the lactococcus 22%. Now, if you see the unique bacteria, like although we know that starter cake and the rice beer are the two similar compounds which are like comprehending each other, but they both have the unique bacteria also, like which are not found in both together. Like you can see duodenobacillus and here alkaliphilus bacteria, which are not common. And among the common, we can see it is in the down table. You can see this is a lactobacillus, lactobacillus, bacilli, and lactobacillus. These are the common. So here we can see that they are connected very properly, and there are various kind of microbes which are not matching us. Now, what are their rules? That one we don't know so far. So this is a Sankey diagram. Here you can see how the microbes, the number of microbes, it came up from family level to genus level, which is the highest number. Here, this is the starter cake where we believe that from here all the microbes came. Here you can see the most dominant, you can see the G part. G is the genus. Here in genus, you can see it is the lactococcus. The lactococcus is the highest. I think my cursor is available. That's why I saw pointer here. You can see this is lactococcus. This is starter cake. But here you can see the lactobacillus, it is very much less. It's not there only. It's less than 1%. But when we come to the final product, here you can see the lactobacillus is the highest number. So this happened in due course of time of fermentation. So now talking about the succession, like how that microbes from which was not there, then it came from some substrate, then it have enhanced to a different level. Here you can see EF2. Here you can see in EF2, the lactococcus was they are in good number, then but in the final product, lactococcus have decreased in number. But lactobacillus was very less in number, but in final product, it have became to 65% in number. This is how the microbial succession happened between the starter cake and the final product. Then you can see in EF1 the number of probiotic and other bacteria. Yes, you can see the number of probiotic bacteria are in very adequate number. Similarly for EF2 also, you can see the probiotic is little bit lesser than other because this is starter cake, not the final product. Okay, now although this is a traditionally prepared starter cake or the rice beer, so definitely there will be contamination and there is a chance of having those pathogenic strain. Then when we check and we classify, we have seen that there are some pathogenic strain, like we have seen like Pseudomonas, Staphylococcus aureus we found maximum. So when we have created a charcoal plot, then we have seen that you can see in this figure, those blue color are the EF1 and yellow color is the EF2. And if you classify them, all those green color radius is all the beneficiary or the probiotic bacteria, and the red color is the pathogenic one. That means very few in number. Okay, it is very few in number because this is a fresh sample. Now, what about if we store the sample for some time, for 6 months, 1 year, what will happen? Can those less number of pathogen can increase to a high number? We was thinking that was our research question. Then similarly, we stored the sample for 1 year, then we will see the next coming up. Then if we check particularly what are those pathogenic microbes in EF1 and EF2, we have seen that those are Pseudomonas, Staphylococcus are there in this number. So they are there, but they are in very less number. That is why it is not harmful while consuming also. Okay, and these are those other pathogenic versus beneficiary bacteria. So it is believed that as it is being prepared by hand and all, so definitely due to hand it have been contaminated those pathogens. Now this is the result of PA1, which is the stored one. Here you can see drastically there is the largest one. You can see this is not a lactobacillus or it is not a probiotic. It is Staphylococcus. This is a pathogenic bacteria. So once the healthy probiotic drink, if you store for 1 year, you will get the result as a very deadly one. It comes around, I think it's around like 27 to 30% of Staphylococcus. And if the species level, if you go, it is Staphylococcus aureus. And when we studied this Staphylococcus aureus, how it is available, it is generally found on human skin and all. So yes, if you see the process, how the fermentation was, how the process was done, it is basically all the women folk have done with their bare hand and all. So it is high possible it came from that part of it. So now the question comes like how will you make it to that pathogen cannot be there, and which level will be the ultimate level that it will have the most amount of probioticness and all. So these are the important question it comes. So if you see the metabolite, yes, definitely you can see the carbohydrate metabolism is highest in both EF1 and EF2. And for PA1, we can see this is the methane metabolism is the highest, of course, because it is degrading. So yes, using NGS technology, this rice beer and starter culture and EF1, we have been sampled, we have sequenced them, and we have observed that starter culture is dominated with lactobacillus, but the final product it's come around with lactobacillus, around 65%. And among the other genus, lactobacillus is the consistent in both the starter culture and the final product. And if you see the probiotic bacteria or probioticness of between EF1 and EF2, it rise from 43 to 68%. So food safety concern come because as we can see prominently Pseudomonas, Staphylococcus, Streptococcus are very much common. So once their population increases or it rises, then definitely this product or this food is not good for health. So here we should we have to develop some new protocols and all how to make those things very properly so that it do not get contamination. So this specific pathogen commonly associated with human skin, as I already told, even a minor sanitation lapse can shift the microbes in upon. So if it get little bit contamination with different types of bacteria, then if you store it for some while, then definitely it's going to change the entire dynamics of the microbes. So the take home message from my entire research is that yes, this is the first comprehensive metagenomic analysis of fermented beverage of missing tribe. Yes, the product have probiotic rich microbes, but despite those probiotic, there also it harbors certain pathogenic strain. And during storage, if the storage is for long time, then rather than probiotic dominance, pathogenic nature enriches. And this study bridges cultural knowledge and modern microbiology, as with this understanding of our research, we can improve the traditional brewing like standard sterilization and controlled fermentation. Thank you. Any question? সেইটোৱেতো জানিবলৈ এটা ডাঙৰ কোৱেশ্চন এতিয়া কোনটো টাইমত বাঢ়ি যায় আমি এটা কি কৰো মেটাজেনমিক্সটো অলপ কষ্টলি হৈ যায় একবাৰ সিকুৱেন্সিং কৰিলে 18000 টকা মান পৰি যায় এতিয়া আমি এক বছৰটো কৰিলে আমি নেক্সট চাব লাগিব 6 মানৰ এতিয়া আমি এক্স্যাক্টলি চাবলৈ কোনটো টাইমত মানে এটা হাইয়েস্ট পিক লগে পায় তাৰ পিছত ডিগ্ৰেড কৰা আৰম্ভ কৰি দিয়ে বা পেথোজেনৰ লেভেলটো বাঢ়ি যায় এতিয়া আৰু এনেকৈ কোৱেশ্চনো আহিব এতিয়া আমি যদি কন্ট্ৰোল কন্ডিশনত যদি কৰো তেতিয়া সেলফ লাইফটো কেনেকৈ হ'ব পাৰে So once you do research, then it comes always new challenges and all. So exactly I cannot tell which is the right number or right numbers of days. Isolation আমি আউটসোর্সিং করছি তাহলে মেটাজেনমিক্স করাই দিচ্ছি সিকোয়েন্সিং করিছে স্যাম্পলটো আমি পঠিয়াই দিছো স্যাম্পল পঠিয়াই দিয়াৰ লগে লগে নতে মেটাজেনমিক্স কৰি পেলাই এভৰিথিং উইথ মানে নতে তেওঁলোকে কৰি দিছে Okay, any question?
Yes. Thank you. For your
Next participant to I will next question. This nang gusto niya ang line of online.
So we thank our men. We thank our men for our time, we will going to retain our session date, and I request our Nandita sir will come here and review this session. Bro. Yeah, I see. Amazon. Oh, no.
Private certificate.
Next 10 points. সহায় করার জন্য ধন্যবাদ এই পরবর্তী সময়টা খুব পান Oh, operator. আমি লাইভ
Same on topic of developments, NPSP ecological plants used by the Boro tribe of Bangsa district, Assam, and this is my content page, which includes introduction, aim and objective, material and method, result and discussion, conclusion, acknowledgement, and reference. So, before going into the topic, I would like to introduce what is asthma. This is a chronic inflammatory respiratory disease that causes recurrent episodes of wheezing, breathlessness, chest tightness, and coughing. According to WHO, asthma affected an estimated 262 million people in 2019 and causes 4,55,000 deaths. So it is a concerned disease which prevail in Assam also. As we know, Assam is rich in biodiversity and indigenous traditional knowledge, and the rural people here are often seen using various medicinal plants for curing different ailments, including asthma. So,
I would like to introduce this presentation here. My aim includes to conduct a preliminary survey on the use of anti-asthmatic ethnomedicinal plants by Boro tribe of Baksa district, and to identify and document the medicinal plants with their indigenous way of preparing the medicine. So we can see here the location map of Baksa district in Assam. So here we can see we surveyed a total of 100 respondents. Among them, male person were 76 and female were 24. The data collection was done through structured and semi-structured interview with the traditional healer, and the specimen were photographed during the survey, and later identification was done through personal expertise and experience, and using books such as Plant of Medicine and Medicinal Plant of Northeast India. And Medicinal Plants of Assam by Sharma UK 2004.
Next slide.
Here, we recorded total of 31 plant species belonging to 20 families. Among them, Solanaceae and Fabaceae emerging as the dominant families, followed by Zingiberaceae, Piperaceae, Lamiaceae, Acanthaceae, Lauriaceae, Fuaceae, Moraceae, Mimosaceae, Brassicaceae, and so on. We found four plant from Solanaceae family and four from Fabaceae family. This is the table of medicinal plant. Here, this column includes the plant with scientific name, the family, the vernacular name, that is Assamese name of that plant, and the use part and the preparation method of medicine. Here we can see the Dotura stromonium, and which belong to Solanaceae family. And
Not to mention again, this is mention the name.
these are also some of the plants. These are the photograph of some plant which were found during the survey. This is Acacia tora. This is also known as Katuriata, and this is Bokful.
You can mention local name.
Yeah. This is the first one is Bon Medelwa, and the second one is Katuriata. The third one is Bokful, and this is Punornava, which is consumed as hak, and this is Giloi, commonly known as Giloi, and this is Bogabaho, Tita Bhikuri, and this is, I have to see the,
It's okay.
Yes.
Next slide.
okay, and this is actually preliminary survey which provides a baseline record of anti-asthmatic ethnomedicinal plants that has been using by the Boro tribe, and this is important for further pharmacological and phytochemical research. Investigations are needed to validate their efficacies and safety, and preservation of this indigenous knowledge is crucial for further advanced research and developing new plant-based therapeutics. That is, This survey is a baseline for research in pharmacological study and in physiological study also. And I would like to acknowledge the herbal medicinal practitioner and traditional healer for their kind cooperation and valuable knowledge and support during the survey. And these are some of the references. And thank you all for listening to this.
Great, and okay, you can for discussion. Is there any question? How I can read as a question?
Regarding these herbal medicines and all of this, I just want to know that actually these are the common. Traditionally, lots of books is there. I think big books are there regarding this medicinal information. But whether this medicinal this herb are scientifically applicable or not, this kind of study is needed. I think.
Yes, sir.
Again, this the medicinal store or this allopathic or this Ayurvedic companies are producing for cure of these asthmas or like this. I think we have to compare these things.
Yes,
That
sir.
study will be, I think, better beneficial. But this kind of study only information. This is the harm. This is the using. This type, I think now it is outdated. I think.
Sir, this is preliminary survey. That's why.
This I think
We need to do some phytochemical analysis to validate their efficacy also.
I think these are already done. And it is not level of survey. I think still it is not level of survey because it is already published. Lots of books is there regarding this medicinal herbs and the bodos. Big book is there where we can get every information about these things. So I think it will be beneficial if we study the effectiveness of this medicine scientifically, whether it is equivalent or not along with this allopathic medicines.
Yes, sir. We will study later.
This will be better. This is my just suggestion. What I observe.
You need to do biochemical analysis.
Yes, sir. Sir, এইটো পিছত মানে এইটো জাস্ট মানে ওয়ার্কার একদম
Yeah, but it was what study very good. Yeah, some people I said I'm not over.
Just it is my feeling. I just sharing.
Sometimes we say it will go turn out মানে study এটা পান উনৰ preliminary হক বা কিবা investigation. এটা মূল প্ৰশ্নটো হৈছে কিমানজন মান তেনেকুৱা এই মানে যিটো ট্ৰেডিশনেল হিলাৰ বুলি কোনা সেইটো কিমানজন মান মানুহ আছে
এনে স্যার around 100 মান জন
প্ৰেক্টিক্যালি কিমানজন পাইছা যে আমি কবিৰাজ বুলি কওঁ যে তেনেকুৱা মানুহ কিমান
যেতিয়া আমি সার্ভে কৰিব গৈছো তেতিয়াতো স্যার মানে around 100 পাইছো আৰু বাকি এনে আমাৰ অসমীয়াত কিছুমান পোৱা যায় কবিৰাজ বা
কবিৰাজ নেকি বুলি
অজা সঁচাকে লগ পোৱা হৈছে নে পাইছে এইটো এশজন অজা লগ
এশজন ওজা পোৱা নাই এইটো যিটো
ডাঙৰ অঞ্চলত এজন
এইটো কমিউনিটিত মানে স্যার সেটা একদম কবিরাজ স্যার 10 জনও পোৱা নাই
আমি তো একটু ইসদাল প্ল্যান পাবলি না মানে স্ট্রিট কোনদাল বেশি করে হয় এনেবা কিবা সার্ভে পয়সা দিতে এইদাল বেশি রেসপন্ড করে একটু ইসদালে হয় কিন্তু ইয়াৰ ভিতৰত এই মিডাল বেশি করে হয়
স্যার এটো যেটো বকফুল আছে হেটো কৰে আৰু তোতুৰটো কৰে
ইমান পার্সেন্ট সার্ভে ডিসমাস এন্ড আদার ডিসমাস কথা দিন চিন্তা করার যোগ বকফুল যদি বকফুলে যদি কিউর হয় তার বকফুলের খেতি করছে নাকি ঘৰত আছে নেকি মেজরিটি অফ দ্য পিপল
সবে যদি ইউজ কৰে ডেফিনেটলি সবৰ কৰে
স্কুপ পাইছে তেওঁলোকে যে বকফুল খালে হয় তো ময়ো ৰুম এডাল কিয় ৰুব এডাল তেনেকুৱা মানে অন্য ধৰণে নলবাই আপকামিং এক্সাম গতিকে আগুৱাই আহিছে আৰু বেছি সোমাই যোৱা নিজে নিজে সোমাই যোৱা ইউনিভাৰ্চিটি এখন পাইছা তাৰে কোৱান্টিটিবোৰ কথা কৈছে বেছিভাগ কোৱান্টিটি পাৰ্চেন্টেজ বা সেয়া ধৰণৰ কাক কৰিব পৰা কিমান ঘৰ মানুহ চাইছিলো তাৰে 70 ঘৰে যে এইডাল প্লান
automatically that percentage that means specifically hetu plant or pora phytochemical koribo nalage তুমি কি ভিত্তিত লবা কোনটো প্লান্ট এটা তুমি phytochemical কৰিম বুলি আৰে কৈছে যে এনেকুৱা ভাবে কৰাতকে অলপ আৰু বেলেগ একো ইমান কাম নহয় এতিয়া phytochemical স্কোৰ কিয়া বহুত তুমি মাসব হেতু আইডেন্টিফাই একদম বেলেগ লাইনত দিছে তারপরে বেলেগ লাইনত দিছে তোমার লাইনতে স্কোপ আছে টাইম আছে স্কোপ আছে সব আইডিয়া দেবেন
So thank you very much for your presentation. Next participant is Omita Boru, Koroma College. The topic is ethnobotanical study of aromatic plants used in culture, culinary and medicinal practices among the Bodo tribes of Pakistan district. Omita Boru, your name is?
Sir,
So you tell your good names. তোমার নাম কই যা কারণ মই তুমি নামটা কই না What news? Mulai dari dia. Toko korai. Dia. Ya ada di kisah. Kalau korai korai kan ngomong di pasar itu. Presentation tahan ya kan? Itu loh itu lagi klik kunci dia buat lo ini. Korai lihat.
slide show না ইয়াত এটা প্রেজেন্ট কৰি অনলাইনে চলি আছে হ'ব এনে
good afternoon. Myself, I am currently working as a guest faculty of Department of Geology, Koroma College. So respected all the faculty members of Tihu College and other institute and my dear students and also my fellow participants. Today I am going to present on an ethnobotanical study of aromatic plants used in culinary and medicinal practices among the Bodo tribes of Pakistan, Assam, India. So moving to the first slide. Sir, slide.
Please take a look at
One is sir, slide.
vibration behind one. এত স্লো হলে তেনেকুৱা হৈ যায়
introduction. So in introduction, so started with what is ethnobotany. Ethnobotany is an interdisciplinary branch of science which occupy all the traditional knowledge, cultural practices, beliefs of human societies, to the use of plants for medicine, food, rituals, and ecological management. Next, moving, what is aromatic plants? So aromatic plants are basically one of important sources of medicinal constituents widely used in human healthcare nowadays. And after that, the antioxidant and antibacterial capabilities of all aromatic plants is advantageous in prevention and reduction of primary diseases. And in case of culinary practices, aromatic plants, traditional cuisines often integrate various aromatic plants, not only for taste, but also it has some therapeutic fluctuation, also reflecting some holistic approach where medicine, arts, and cuisine are closely interconnected. And after that, the Bodo tribes have their own traditional culture, and due to their association with plant communities, they have good knowledge about food and medicinal use of aromatic plants. So moving to the next slide. So here is a map of the study area. The present study was performed in the Baksa district of Assam. So all we have known that the Bodos are found as one of the major tribes of Baksa. So in this whole through survey, we have found about 40 species of plants, but due to lack of time, it is not possible to discuss here. So we have choose some specific plants that have highly medicinal and aromatic value. So here, the first species, the botanical name of this species is Pomalomena aromatica, which belongs to the family Araceae, and the life form of the species is herb. And basically, the leaves of these plants are used, and the basic uses of these plants are that is used in fish fermentation due to its a good aromatic properties, and as it enhances the aroma of all the fermented fishes. So moving to the next slide. The botanical name of this species is Calacosia esculenta, and it belongs to the family Araceae. The life form of the species is herb, and basically leaves and stems are used. And in tradition, basically in Boro tradition, during the fish fermentation process, due to its aromatic properties, it enhancing all the aroma. Also, it's prevent all the spoilage, and it reportedly used to treat some conditions associated with low blood levels. Here is another species name. The botanical name of this species, Andrographis paniculata, which is belong to the family Acanthaceae, and the life form of this species is herb, and basically leaves and the stem of these plants are used. So in our tradition, the extracted juice of this plant consumed by human to treat various like intestinal worms, bacterial infection, and also control diabetic condition.
Just mention the name and inches. Not to mention the family.
So here is a species. The botanical name of this species is Anhydra fluctuans, and the uses of this plants is target of every Bodo tribes in our Bodo cultures. It has been reported that It is a species used to treat various skin disease associated with all kinds of rashes, itching, and it also improving all eyesight problems. So here is another species. The botanical name is Bryophyllum pinnata, which is traditionally used to treat kidney stone and also urinary tract problem. Another species, plant species, which is Cotonia cordata, and it is used in various culinary diets such as all kind of chutneys, salads, and meteor fry fish curry. It has also anti-properties to maintain stomach pH level. So here is another species. The botanical name is Leucus aspera. It has been traditionally used to treat the sinus problem and also treat it in allergies, and also extracted juice of this species used to treat epistaxis, which is belongs to the nose bleeding condition. Here is another species which is Osima stenothorum. So traditionally in our Bodo, along with our Hindu religion, it plays a significant role in Hindu rituals along with worship, and also it reportedly used to treat all cough, common cold, and also the extracted juice is applied on wounds, ringworm, etc. So it is another species named Citrus orangifolia, which is traditionally used in all Hindu rituals as well as therapeutic application in treating all kinds of skin disease, boosting immune function, and it is used for making various kinds of pickles and adding flavor to various dishes. Another species name, which is Eryngium foetida. This plant species is traditionally used to enhance the flavor, the aroma of various kinds of curry or dishes in local culinary preparations, such as all kinds of chutneys, through adding depth and some complexity to traditional recipes. Another species, which is Hydrocotyl rotudifolia. This leaf is traditionally consumed to treat respiratory issues like pneumonia, gastrointestinal problems such as stomach issue, dysentery, and also incorporated into the local culinary type of Bodo. Here is another species named Paideria foetida, and this species is traditionally used to treat condition associated with low blood levels, weakness, and various kinds of stomach issues. So here are some references. So my own study is contribute to understanding of the relationship between all Bodo community and the aromatic along with the medicinal plants, and it emphasizing the need for conservation, also documentation, sustainable use, and incorporating traditional practices with modern healthcare and cuisine. So here is my overall presentation. I hope all of you understand, and I think that you may gain a little bit of knowledge through this presentation.
So the topic is open for discussion now. It is a review paper, nothing.
মানে স্যার এটো মানে জাস্ট মানে কালেক্ট কৰি থকা হৈছে, মানে আমি কেইবাটাও স্পেচিছ পাইছিলো।
How done practically?
নহয় স্যাৰ, এটো মানে বডো ট্ৰাইবছৰ মোৰ যিজন কোথৰ আছে তেওঁ মানে যেহেতু বডো ট্ৰেডিশ্যন, তো তেওঁ আৰু মই লগ লাগি মানে বডো বিলাকৰ ধৰক ঘৰৰ পৰা বা ডাঙৰ মানুহৰ পৰা আমি ফুটি লৈ এইখিনি তাৰ মানে কালেক্ট কৰিছো। হয় স্যাৰ, ফটোগ্ৰাফবোৰ নিজৰ, তাৰ মানে কেইখনমান ফটো মই নিজৰ ঘৰৰ পৰা উঠাইছো, আৰু বাকী কোথৰ জনে কালেক্ট কৰিছে। কিছুমান ধৰক তেওঁ নিজৰ ঘৰৰ পৰা বা ওচৰৰ চুবুৰীৰ পৰা, আৰু বেছি সংখ্যক ফটো মই ঘৰৰ পৰাই লৈছো স্যাৰ।
যে এটা ইনফৰমেশ্যন দেখুওৱা হৈছে যে পাতে গোজা নাকি দেখাইছিলে, সেইটো দেখুওৱা সময়ত কোৱা হৈছিল যে এইটো কিডনী পাথৰৰ কাৰণে ব্যৱহাৰ কৰা হয়। এনেকুৱা কোনোবা ইনফৰমেশ্যন দিছে নেকি যে কিডনী পাথৰৰ কাৰণে এনেকুৱা ধৰণৰ বেমাৰ চাৰ্ভে তেনেকুৱা ধৰণৰ ইনফৰমেশ্যন পোৱা গৈছে, না ক'ৰবাত ৰেফাৰেঞ্চৰ পৰা লিখা হৈছে?
মানে
সার্ভে করার সময় মানে যেতিয়া সুবিধা হৈছিলে মানে বড়ো কমিউনিটিত তেওঁলোকে এইটো কন্ডিশন
পাথৰৰ কথা কৈছে
কৈছে স্যার মানে এইটো কন্ডিশনত ব্যৱহাৰ কৰা
আমি পাইছো এইটো কাৰণে সুধিছো আচলতে কিডনি পাথৰৰ যিটো ইনফৰমেশ্যন নৰ্মেল ইনফৰমেশ্যন এইটো ৰিচেণ্ট আগতে এইটোতো নৰ্মেল ইনফৰমেশ্যনে নাছিলে বা মই বিলংছ টু বড়ো কমিউনিটি so I am asking so there is no information about these things এইটো যদি ক'ৰবাত এনেকুৱা ইনফৰমেশ্যন দিয়া যদি কোনোবা মানুহ ওলাইছে তেতিয়াহ'লে এইটোতো অদ্ভুত কথা হৈছে মই এইটোৰ কাৰণেহে সুধিছো আচলতে
বড়ো লগত কমিউনিটিটো আইডিয়া নাছিলে থাকে কে জানে বা বেলেগ ট্ৰাইবে জানে
মই তালে সুধিছো যে বাস্কাৰ ডিষ্ট্ৰিক্টত এনেকুৱা ইনফৰমেশ্যন দিয়া মানুহ পাইছে নেকি তাৰমানে না ক'ৰবাত ৰেফাৰেঞ্চৰ পৰা দেখিছে এইটোহে পাইছে তাতে মানে পোৱা গৈছে তেনেকুৱা মানুহ প্ৰেক্টিকেলি তুমি সেইটো পাইছা প্ৰেক্টিকেলি undergraduate still studying my other so they are undergraduate student oh okay thank you Sandra for your presentation so next participant is Dipanvita Das Empress okay you can go now